My name is Florian Eichler. I'm director of the Center for Rare Neurological Diseases at Massachusetts General Hospital. The title of my talk is Rare but Mighty: Scaling Up Success in Single Gene Disorders. I'm a neurologist who sees genetic diseases, and in my experience, the number of rare single gene disorders is increasing in large part due to diagnosis by next-generation sequencing as well as newborn screening. And at the same time, we're seeing an increase in the technologies for gene correction, whether it's in vivo gene therapy or ex vivo gene therapy. And many families and patients are coming to me today asking how to make progress, how to go from diagnosis to treatment, and how to initiate this whole process. In my mind, the large problem today is how to engage and how to know what cells to target, what biomarkers to follow, and how to make this widely available and scale it up for patients in need. Within the Center for Rare Neurological Diseases, we try to address these gaps. The center lives at MGH and Harvard and MIT in this interface of the landscape. And we try to here understand that in different diseases, there are different needs. One disease might be missing a natural history component. There might be outcome measures or an industry partner missing in another. Within my lab, we are addressing biomarkers and trying to understand the connections to the relevant cell types in the diseases. We use lipid chemistry and combine this with cell and animal biology in order to understand how to get the right target cells in gene correction and how to really successfully scale up. To give you an example of one of our early successes, here an old lipid disorder known as adrenoleukodystrophy that became famous through the movie Lorenzo's Oil. What we discovered is that what was critical for this brain disease was a certain type of inflammatory cell called a microglia. And if we could correct those microglia by performing gene correction on the precursor cells in the bone marrow, we would be able to salvage and rescue this progressive disease. And we were able to do this using an ex vivo lentiviral gene therapy on the boy's own cells, thereby avoiding engraftment problems and graft versus host. Today, boys that previously used to come to me at death's door and in wheelchairs now are reporting back to me from their summer camp and living fulfilled lives. Another example of a lipid disorder that we tackled is rare familial neuropathy due to mutations in SBTLC1. This gene encodes an enzyme called serine palmitoyl transferase. And what this enzyme does is that it makes the enzyme promiscuous to its substrate, so that instead of taking L-serine, this enzyme now starts taking alanine and glycine and making a toxic deoxysphingolipid that causes painful ulcerations and amputations in these patients. So we figured out that we could flood the system with L-serine and thereby reduce these toxic deoxysphingolipids. And in a placebo-controlled, double-blinded, FDA-funded trial, we were able to improve neurologic function. And as you see in the bottom left here, in some patients, we even saw regrowth of nerves in the skin, where for years this neuropathy had destroyed the small peripheral nerves. But not all mutations in this enzyme act this way. And what we discovered is that there were some mutations that did not lead to a rise in deoxysphingolipids and caused a problem to development in children. And here you see two young girls affected by mutations in a small subunit of this same enzyme. And what happens here is that because of the mutations in this small subunit, the sphingolipid production cannot be properly regulated and cannot be shut down, as shown in the bottom left corner here. And because of that, these girls have an excess in sphingolipids, and their enzyme, so to speak, lacks the brake pedal. And so we're working right now on how to fix that and improve their brain development and regulation of serine palmitoyltransferase. So in short, what we are showing here is that we're opening doors to new biology by examining individual genotypes. We show that even mutations in the same gene, such as SBTLC1, can on the one hand give rise to toxic deoxysphingolipids that harm sensory neurons, but other mutations do not lead to those kinds of deoxysphingolipids, but cause an excess of sphingolipid production that harms motor neurons. On the other hand, we see mutations in genes such as ABCD1, where not only lipids are affected, but also neurotransmitters. And this is giving us a new path into biology that we previously had thought to be about lipids alone. So what are the next steps and what are opportunities? I think we are really on the verge of developing cell type specific biomarkers that are going to help us scale up and move more quickly and more successfully from bench to bedside and employ the tools of gene editing and gene therapy by understanding these specific biomarkers that link to neurologic function. So today, when families come to me and are asking for treatments that are targeted and that are successful and that can be employed quickly, I speak with more confidence than just 10 years ago and think we can do this better than before. But to do this, we need partnerships. We need to work together with industry across different stakeholders and scale up by expanding and working together.